Let's start our chapter 5, which is about proteins. In this chapter, we will talk about the chemical structure of protein, how proteins are digested, absorbed, and transported. We will talk about the protein metabolism and muscle protein turnover, nitrogen balance, and we will talk about the recommendations for athletes regarding protein and so on and so forth. This chapter is a lot and covers so much information. So, let's start with the structure and function of proteins. As you know, protein they are made up of amino acids, and amino acids are linked by peptide bonds. And from peptide bonds, we get polypeptides or proteins. So, amino acids are the building blocks of proteins. And here we have the chemical structure of an amino acid. Each amino acid has an amino group, an acid group, and a side chain. And this side chain is different for each amino acid. And we have 20 different amino acids, and those 20 different amino acids they differ in terms of the side chain or side group. Let's do this. It's kind of introduction. So dispensable amino acid, indispensable amino acid, and conditionally indispensable amino acid. In the past, they were referred to as essential or non-essential amino acids. But today they call dispensable amino acid for non-essential amino acids. So they can be manufactured in the liver, and also we have six of the dispensable amino acids. So number one is A. So dispensable amino acids are the amino acids that are non-essential. And when we say non-essential amino acids, it means they can be manufactured in the liver. Indispensable amino acids, we cannot make them. These are essential amino acids. And the conditionally indispensable amino acids, these are the amino acids that cannot be made during periods of physiological stress, like injuries, different disease, and prolonged exercise. So the liver is not capable of making those amino acids under some conditions, and those are called conditionally indispensable amino acid. So based on that, we can divide amino acids into indispensable, dispensable, or conditionally indispensable. We have 20 amino acids. Nine of them are indispensable, which means these are essential. The body cannot manufacture them. 11 of the 20 amino acids are dispensable. The liver manufacture them. And six amino acids are conditionally indispensable. These are six amino acids out of 11 dispensable amino acids. So during the periods of stress, body cannot make them, like in illness, injury, prolonged endurance exercise. Here is a summary of the 20 amino acids we have. Um, you know, uh, as a dietitian, you need to know these names, but you don't have to remember or memorize the chemical structure of each amino acids. But here is a very important point in this table. For example, for the alanine, we have glucogenic and ketogenic. So alanine is glucogenic, but it's not ketogenic. So here is the important term here. Glucogenic, it means these amino acids can be transformed to glucose, or like the liver can make glucose out of these amino acids. So these are glucogenic. Ketogenic means they cannot make glucose, or they can be converted to fatty acids. That's called ketogenic. So for instance, isoleucine is both glucogenic and ketogenic, which means these can be converted to glucose or ketone bodies or fatty acids. And here we have other amino acids. So, and also we have classification here, like each amino acid is whether indispensable or dispensable. And here's the chemical structure. Again, you don't have to memorize these chemical structures, but you need to know at least like the simplest amino acid is glycine, and the R side group, R side chain is just one atom of hydrogen, and the alanine is the second simplest one, and the side chain or side group is a methyl. And it can get complicated. Like, for example, you look at number 19, it's tryptophan. It has two rings, so it's complicated compared to other structures, other amino acids. Deficiency, amino acids, some people can develop deficiency in amino acids. We have like frail elderly people in liver and kidney disease, they can develop deficiency or food insecurity, eating disorders. So this can cause deficiency. And the most important indispensable amino, the most important essential amino acids. It's better to remember these amino acids. We have lysine, threonine, methionine, and cysteine. These are the most important essential amino acids. We need to get them through diet. So our body, specifically the liver, cannot manufacture them. That's why they are called indispensable amino acids. Protein quality is a very important term in nutrition. It's determined based on the amounts and types of amino acids and their digestibility. So there are two criteria to determine protein quality. Each food item, for instance, beef, egg, wheat, we can categorize them based on protein quality. And in order to determine protein quality, we use two factors. One of them is the digestibility of the protein. It means like how much of that protein our body can digest and absorb, and also the amount of the essential amino acids, whether they convey all essential amino acids to the body or not. So the FAO, the Food and Agriculture Organization, they develop different methods to determine and measure protein quality. We have DIAS and PDCAS. And there are other methods, but these are like old methods, like biological value and protein efficiency ratio. So for the PDCAS, it stands for Protein Digestibility Corrected Amino Acid Score. It's a scale from 0 to 1, and it's based on protein absorption, utilization, and amino acid content. Another method is DIAS, which stands for Digestible Indispensable Amino Acid Score. It assesses the ileal versus fecal digestibility. It can be greater than 1, so it's not necessarily should be between 0 to 1. It can be greater than 1. And the higher score means better quality. So again, we can categorize protein food sources based on these two methods, based on the protein quality. Like for instance, here we have beef, casein, egg, milk, whey protein, soy protein, rice, peas, and wheat. As you can see, the animal-based or animal-derived food sources, protein food sources, they have higher protein quality. Because the animal-derived proteins, they contain all essential amino acids and they have higher digestibility compared to plant-derived food like rice, peas, and wheat. They have lower PDCAS and DIAS scores. When we say complete proteins, it means the food item or the protein we get through diet contains all the indispensable amino acids in proper concentration. The complete proteins like the beef, milk, casein. Casein is a protein in the milk in dairy products. They support growth, they prevent amino acid deficiencies. And incomplete proteins, they lack one or more of the indispensable amino acids. For instance, rice, if we just eat rice, just plain rice, it's incomplete protein. It lacks some essential amino acids, some indispensable amino acids. Complementary proteins, it's like pairing plant proteins and combining two incomplete proteins. For instance, if we combine rice with legumes, we have some limiting amino acids in legumes and in rice. And if whenever we combine them, we can get all the indispensable amino acids. This is what we see like in Mexican food. Like they serve rice and black beans. And black beans is a legume. So by eating rice and legumes together, we are kind of doing a complementary proteins like we are getting all the indispensable amino acids. And here is the proteins in different foods. Milk, the milk or most of the time we can say dairy products like yogurt, cheese, they have between 8 to 10 gram per serving of protein. And oils, it's less than one. Fruits is less than one. Vegetables are like between two or three, like carrots, corn, broccoli, and etc. Peptide, again, like amino acids are the building blocks of proteins and we need to link them by peptide bonds. So peptide are two or more amino acids that are linked. Dipeptide is like two amino acids that are linked. Tripeptide, three amino acids that are linked. And polypeptide is proteins, like they have more than 10 amino acids which are linked together. And the function of protein, we have structures of protein, different levels of structures, primary, secondary, tertiary, and quaternary. The primary structure determines the function of protein. And here, this table summarizes the different um, functions of proteins. It's better to note here when we say protein, protein means of prime importance. That's why we call them proteins of prime importance. They do like they conduct very important tasks in our body, from being an enzyme, hormone, structural proteins like skin, hair, nails. We have collagen like myosin, actin. We have transport proteins. We need proteins to absorb nutrients in the small intestine. We have immune system proteins like antibodies. They are all proteins. We need proteins to acid-base regulation. Like otherwise, we will have more acidic or basic. Like you know, like we will see a disruption in pH if we don't have enough protein in the bloodstream. We need protein for fluid regulation. Otherwise, we can develop edema. We can burn proteins as a source of energy. So they do different tasks. Here we have arginine. Arginine is an amino acid. It stimulates killer cell activity, so it is part of the immune system, and it's reducing the pro-inflammatory cytokines. Glutamine is the most abundant amino acid pool, We will define amino acid pool later, but remember, this is a very important point. Glutamine is the most abundant amino acid. The major site of synthesis is in skeletal muscle. Major fuel source for certain immune system cells and glutamine supplementation. The based on the research, the supplementation of glutamine, it improves markers of fatigue without performance improvement. So glutamine supplementation doesn't improve exercise performance, but it improves markers of fatigue. And here let's talk about the digestion, absorption, and transportation of proteins. The digestion of protein, it starts in the stomach. We have hydrochloric acid in the stomach. It denatures the protein. And when we say protein denaturation, it means like the change in the chemical structure of protein by chemical or mechanical means. So in the stomach, hydrochloric acid or our stomach acid, it does protein denaturation. And also we have pepsin. Pepsin is an enzyme. It breaks the bonds of leucine and tryptophan. So whenever in the peptide chain, which is a chain of amino acids, which are linked together. So uh, pepsin is an enzyme in the stomach, and it breaks the bonds between leucine and other amino acids, and also tryptophan and other amino acids. So remember we don't have any digestion in the mouth, unlike carbohydrates. The digestion of proteins starts in the stomach. And the dietary proteins and large polypeptides, they are broken down to smaller polypeptides. And the amino acids are absorbed via energy dependent active transport like glucose or galactose. For regarding the carbohydrates, we need energy to absorb those nutrients. We need energy to absorb amino acids in the presence of sodium and hydrogen ions. And the amino acids leave the cell by various passive carriers and enter the bloodstream. And from the bloodstream, it goes to the liver via the hepatic portal vein. Two-thirds of the amino acid absorbed in the form of dipeptides or tripeptides. It's very important. One-third of the amino acids absorbed are in the form of free amino acids, but two-thirds are absorbed in the form of dipeptides or tripeptides. More than that, our intestinal cells are not capable of absorbing large peptides. And the liver monitoring and dictating, it has a monitoring and dictating role. Kind of liver decides whether this amino acid should go to the bloodstream and being transported to other tissues or not. And we have exceptions regarding the BCAA. BCAA stands for branch chain amino acids. And we have three branched chain amino acids. They are valine, leucine, and isoleucine. We have low levels of BCAA transferase in the liver, which is needed to transfer these amino acids to tissues. And the branched chain amino acids are taken up by skeletal muscle cells. That's why I know you already heard of like taking BCAA or branched chain amino acid supplementation by different athletes. There's a reason because of this, like BCAA are taken up by skeletal muscle cells. And BCAA transferase can be found in the heart, adipose tissues, and kidney. We have amino acid pool. Remember, I told you like we are going to define amino acid pool later. So amino acid pool is the free amino acids in the blood and the lymph. And it always in flux, which means we have constant change. Also, it refers to as protein turnover. Amino acid pool contains 150 gram of amino acids, of which 80 gram, around approximately 80 gram of amino acid pool is glutamine. So remember, we mentioned like glutamine is the most abundant amino acid in the amino acid pool. So 80 out of 150 gram of amino acid pool is glutamine. 10 to 20% of resting metabolic rate each day is expended on synthesizing and degrading proteins. So we have amino acid pool and protein turnover. So protein has two sources in the blood in our body. It can be exogenous or endogenous. So exogenous is a dietary protein or amino acid supplements. These are the proteins we get through diet. Or we have endogenous protein source. These are the dead mucosal cells, enzymes, and other protein containing secretions. We all absorb these amino acids, and in the blood and the lymph, we have amino acids. It's called amino acid pool. So liver and muscle, they take up the amino acids from the amino acid pool. And also we excrete amino acid through skin. Like we shed skin each day and we lose protein through that. Or kidneys as urine or colon as feces. So we lose amino acids. That's the excretion. So in this picture shows the protein turnover. So we are getting protein, we are absorbing protein are taken up by different tissues and we excrete it and then again we use it. So we have a flux, constant change in the amino acid pool, which is called protein turnover. Some of the amino acids have recently been absorbed from the gastrointestinal tract, but most come from the breakdown of body tissues, including skeletal muscle tissue. And again, as we said, 80 gram of the 150 gram of amino acid pool is glutamine. And we have more dispensable than indispensable amino acids in the amino acid pool, and it's always in flux. Let's do this review. You can pause the video and do it on your own and then resume the video. So let's match these on the left side with the terms on the right side. Mucosal cells shed in GI tract. So that's an endogenous protein. Branched chain amino acid supplement, that is exogenous protein. Hard-boiled egg, that's exogenous protein. And enzymes, these are in endogenous protein. And let's talk about the protein, the metabolism of protein and amino acids. Remember, the liver is the major site of metabolism of protein. Other tissues also do protein metabolism, but the liver is the major site or the major organ or tissue to do protein metabolism. Proteins, they have two metabolic processes, deamination and transamination. When we do transamination, specifically in the liver, we are making dispensable amino acids from indispensable amino acids. Here in this picture, we have deamination. Transamination means getting amino group from one amino acid and transfer it to another structure or chemical substance and make it an amino acid. So it's like transferring amino group. But when we say deamination, it means like removing the amino group from an amino acid. So here in this figure, we have deamination. We have a threonine, and when we do deamination, we are removing the amino group And by doing that, we are making alpha ketobutyrate, which is a kind of a keto acid, and ammonium. And this ammonium will be excreted through urine in the kidneys. Um, so this is deamination. Liver take up amino acids from the bloodstream, or it make amino acids for different reasons. So we have protein anabolism, which means making protein. Liver enzymes, these are proteins. Liver makes plasma proteins like albumin. Albumin is a very important protein in the bloodstream. It carries calcium, free fatty acids, and etc. Liver synthesize creatine from arginine, glycine, and methionine. And also, it provides energy to the liver cells. The source of 50% of the ATP used by the liver comes from amino acids, like almost half. And the muscle take up amino acid from the bloodstream, from the amino acid pool, and it can oxidize it, can synthesize proteins to build muscle, which are called anabolic, or catabolic, which means breaking down proteins and transfer it to the amino acid pool. So it's called protein turnover. Why protein turnover? Because muscle take up amino acid from amino acid pool and make protein, or it breaks down protein and oxidize it, or give it to the bloodstream. It's called turnover. It's kind of a circle. So amino acid they undergo deamination and transamination, which leave us with carbon skeleton because we are removing the amino group. What we are left is carbon skeleton or alpha keto acid. We can use alpha keto acid as an energy source to produce ATP. And the amino group as ammonia or ammonium ions, we need to get rid of these. We need to get rid of ammonia or ammonium because these are toxic. So we make urea in the liver and they excrete urea in kidneys, in urine. And here we have the Krebs cycle. The important thing about this figure is this. The structure of the amino acid determines the entry point into the Krebs cycle. So here on this picture, as you can see, some amino acids, they are converted to pyruvate, and from pyruvate it goes to the Krebs cycle. But some amino acids, they can be directly entered to this Krebs cycle without being converted to pyruvate. So the structure of the amino acids determines the entry point into the Krebs cycle. And 3 to 5% of the total energy in our body comes from amino acids on a daily basis. And these are the energy yielding amino acids in muscle cells. So we have branch chain amino acids, asparagine, aspartate, and glutamate. These are the main amino acids that muscle cells can use as energy source. And we have adrenal glands. These are like very tiny glands on top of the kidneys, and they produce cortisol or stress hormone. And this cortisol, it promotes proteolysis, which means breaking down protein. In endurance exercise, which is an acute stress. So in acute stress, we have cortisol or stress hormone. So we have breakdown of proteins for energy in muscle cells. Or in the prolonged endurance exercise, we have increased oxidation of leucine. Leucine is one of the branch chain amino acids. And this metabolic response is affected by the carbohydrate content of the diet. This is a very important point. I don't want to go into detail about this picture, about this figure, but remember, alanine, which is the second simplest amino acid we have, is not used by the skeletal muscle. Alanine enters to the blood, it goes to the liver, and in the liver it can become pyruvate, and from there it can become glucose. So we have gluconeogenesis. And nitrogen balance and net protein balance. These are very important points, but very simple. So nitrogen balance is determined over several weeks. We are losing amino acids or protein through urine and feces. And in growth state, we have positive nitrogen balance. So nitrogen balance is like total nitrogen intake minus total nitrogen loss. So it's like a mathematical problem. So nitrogen balance, whenever total nitrogen intake is more than total nitrogen loss, so we have positive nitrogen balance. Um, if the total nitrogen intake, which means getting protein through diet, is less than total nitrogen loss, then we have negative nitrogen balance. So net protein balance, it means muscle protein synthesis equals muscle protein breakdown. In positive nitrogen balance, muscle protein synthesis is more than muscle protein breakdown, this is the condition we see in growth period, like in infants, in children, in adolescents, or pregnancy, in hypertrophy, increase in skeletal muscle size. And in starvation, in fast and growing cancers, in fasting conditions, or like semi-starvation, we have negative nitrogen balance. To achieve positive nitrogen balance, adequate amount of protein intake and energy is needed. Let's match these on the left side with the right side. Again, you can pause the video and try on your own and then resume watching the rest of the video. So when we say anabolic or catabolic reactions, so anabolic reactions means building something from smaller parts. Catabolic reactions, which is meaning breaking down something. So catabolic reactions like breaking down of complex molecules. That should be F. Or liver manufactures dispensable amino acids. Is it anabolic or catabolic? So making something from smaller parts. It should be building complex molecules. So anabolic is C. Transamination, which means amino group is transferred to another carbon skeleton. Deamination, which means removal of the amino group. The option E, which means leaves an alpha keto acid or carbon skeleton, that should be deamination. Liver manufactures dispensable amino acids, so it should be transamination. So in the next part we will talk about the protein recommendations for athletes.